All right, next up we want to talk about meiosis. So for meiosis, we're going to see specific cells involved in this process. We're going to see gametes, eggs and sperm cells. Now these cells are very different from somatic cells in that these cells are going to be haploid, meaning that they only have one member of each homologous pairs of chromosomes. So since we have 46 chromosomes total, if we only have one from each pair, we only have 23 chromosomes in our haploid cells. Now the haploid cells or our gamete cells are going to be formed by a very specific process known as meiosis. In the case of meiosis, we're actually going to see two divisions that are going to occur, and we're going to end up, instead of 4 cells, instead of 2 cells, we're going to end up with 4 daughter cells. These 4 daughter cells are going to be haploid daughter cells. Now, there are a couple of different functions of meiosis. The goal of meiosis is going to be to keep the number of chromosomes in a body cell constant from one generation to the next. If we did not undergo meiosis, if we did not undergo the transition from a diploid cell to a haploid cell, then each generation would double in its number of chromosomes through fertilization. So what happens is you have diploid cells. You have 46 chromosomes, your partner has 46 chromosomes. You're going to take that number, divide it in half, so you're going to go from 46 chromosomes to 23 chromosomes. Your partner will do the same, go from 46 chromosomes to 23 chromosomes, and then when you have the fusion of those two gametes to form a zygote, you get 23 chromosomes from mom, 23 chromosomes from dad, to end up with a grand total of 46 chromosomes in total. So that's going to be the process of meiosis. Meiosis is going from that original 46 down to 23. So this is going to keep that number of chromosomes in the body cell constant from one generation to the next, but it's also going to increase genetic variability in the population because you're only getting half your chromosomes from mom, half your chromosomes from dad. Thus, you have a unique genetic combination that neither mom nor dad has. So this is how we introduce genetic variability. And there's actually a couple of ways that we introduce genetic variability in meiosis. It's really exciting. I enjoy this section very much. So we're going to talk about genetic variability quite a bit a little bit later on. So we're going to first give you an overview of meiosis. Meiosis has 2 cellular divisions. Meiosis 1 is going to separate the homologs. Meiosis 2 is going to be responsible for separating those sister chromatids. Meiosis 2 is going to look almost exactly like mitosis. So meiosis 1, we're separating homologs. We're going to go from 46 chromosomes down to 23 chromosomes. And then meiosis 2 is going to separate those sister chromatids. Each division, each phase of meiosis 1 versus meiosis 2 is going to have its own version of prophase, metaphase, anaphase, and telophase. And we'll go through each one of those as we move forward. So starting off with meiosis 1. Meiosis 1 is going to be preceded by interphase. Interphase, take a moment here. Does anybody remember what happens in interphase? Interphase is going to be where the cell carries out its normal cellular processes, but it's also going to be the stage during which we're going to undergo genetic replication. We're going to undergo that S stage where we're going to replicate or synthesize new DNA. During this time, each chromosome is going to be copied and is going to consist of 2 attached chromatids, or 2 sister chromatids bound up next to each other. This is called reduction division because we're going to be resulting in the production of 2 haploid cells. We're going from a single diploid cell to 2 haploid cells in meiosis 1. That's the end goal of meiosis 1. We start off with 1 diploid, we're going to end up with 2 haploid. Each daughter cell is going to have 23 chromosomes, 1 member of each pair of homologous pair. And each chromosome is going to consist of 2 sister chromatids. So all we are doing in this first stage is going from a diploid cell to a haploid cell. We start off with, in this case, in our example here, we have 4 chromosomes. We're going to end up with 2 cells that each have 2 chromosomes at the end of it. Meiosis 2 is where we take those chromosomes and we separate those sister chromatids. So we're going to take those 2 chromosomes here, and we're going to separate them out into we're going to separate those sister chromatids into daughter chromosomes. This should look exactly like mitosis, exactly like mitosis. And we will break down each one of these stages a little bit later on. But first, I want to talk about the interesting aspects of meiosis, specifically meiosis and genetic variation. Genetic variation is going to be introduced a couple of different ways. This is pretty exciting. So it's going to be introduced a couple of different ways during the stage of meiosis. The first way is going to be through prophase 1. During prophase 1, we're going to see that the chromosomes are going to condense down, but they're also going to pair up. Unlike in prophase 1 of regular mitosis, we are going to see homologous chromosomes pair up together. So they're going to find their mate. The 2 chromosome 1s are going to find each other. 2 chromosome 2s, they're going to find each other and they're going to pair up. Now here's the interesting thing. When they pair up, they start swapping DNA. Yeah, it's pretty cool. A little bit gross. They start swapping DNA. They form what is known as a synapse. They're going to line up together. So we have 2 chromosomes here. So this, I know it doesn't really look like it, but let's pretend that these 2 sister chromatids are attached right here at the centromere. 2 sister chromatids here are going to be attached to the centromere. What happens is when these homologous pairs line up, they start swapping DNA. So what you might end up with is 1 sister chromatid that is completely, so here I'm just going to start using people in my family because it helps me understand and keep everything straight. So this would be like if my cells were getting ready to undergo meiosis and they were to line up and pair up, we could have one entire strand of my DNA that is made up of only paternal DNA. This entire strand, this entire chromosome 1, is only made up of DNA coming from my dad. This second strand could have a portion up here that's my dad's, could have a portion right here that's my mom's, could have a portion right here that's my dad's. Then I could have, I will also have another sister chromatid that has mom, dad, mom, and then another sister chromatid that's just my mom. So if you were to think about this in terms of my offspring, I could have one child that has only DNA from my dad. One child that has a mixture of DNA from my dad, my mom, and my dad. One child who has a mixture of DNA that is my mom's, my dad's, my mom's. So in this case could be my dad's finger length, my mom's hair color, and my dad's eyes. This one would have my mom's finger length, my dad's hair color, and my mom's eyes. And this one would just look exactly like my mom. Now this is only for 1 chromosome. Keep in mind, I've got 23 pairs of chromosomes that can organize however the heck they want. But in terms of chromosome 1, I could have 4 completely different daughter cells. 1 cell that contains only my paternal DNA, 1 cell that contains a mixture, another cell that contains a completely different mixture, and 1 cell that contains only my maternal DNA. And that's assuming that we synapse like this. These 2 could have also swapped DNA. These outer cells, these outer ones could have swapped DNA. These ones could have swapped several times. They could have crossed over only once. But that's a crossing over event. This occurs during prophase 1 of meiosis 1. Another introduction of genetic variation occurs during metaphase 1. Metaphase 1, so if you remember way on back when we talked about mitosis, we talked about metaphase. During metaphase, our chromosomes are going to line up down the center of the cell. Now, when they line up down the center of the cell in mitosis, they're all going to form a single line. So we're going to have 46 chromosomes, 1, 2, 3, 4, all the way down the line, 46 chromosomes in a straight line. So they're going to line up single file in mitosis. In meiosis, they don't line up single file. They line up in those homologous pairs. Now, when they pair up, they can pair up in whatever way they just happen to line up. There's no organization. So they could line up with all of my maternal DNA on the left and all of my paternal DNA on the right. They could line up 2 maternals on the left and one paternal on the left, and 2 paternals on the right and 1 maternal on the right. They could line up every other. They could line up 1, 2, 2, 1, 2, 1, 1, 1. They could line up in any way. So the maternal or the paternal member could be oriented to either pole when they line up, when that single cell lines up. So for one cell, let's just say that one cell had only 3 pairs of homologous chromosomes. Now, obviously, we have more than that. We have 23 pairs of chromosomes. But let's just say that we only had 3 pairs of chromosomes. There are 8 different potential outcomes for how those cells could divide up and line up. 8 potential offspring outcomes that are all going to lead to genetically unique individuals. Now, this looks very similar because you're not looking at the crossing over events, but crossing over occurs and then we get independent assortment. This process here of how they line up is what's known as independent assortment. We're going to talk quite a bit about independent assortment when we talk about our genetics, when we talk about the inheritance of DNA and different genetic factors and human variation. All of this will come up again. But for now, I want you to know that synapsis occurs during prophase 1. That's one way to introduce genetic variation. And independent assortment occurs at metaphase 1. This is another way in which we can get genetic variation. Now, the remaining stages of meiosis do not involve genetic variation. In anaphase 1, the homologous pairs of those chromosomes are going to separate. They're going to move towards opposite ends of the cell. Each homolog is going to consist of those 2 sister chromatids. So like I said earlier, we're just going to be separating those, right here. We're just going to be separating those homologous pairs. So we're going to separate those homologous pairs during anaphase 1. And then in telophase, a nuclear envelope is going to form around those chromosomes at the end of each cell, and cytokinesis is going to occur, and we're going to end up with 2 haploid cells. And boom, we have completed meiosis 1. Now, we're not quite done yet, because although we have separated the homologous pairs, we still need to separate the sister chromatids. So we're going to undergo a very brief interkinesis. Interkinesis is a bit like the period before meiosis, it's a brief phase before meiosis 2 that's similar to interphase. Now, it's going to be a little bit different in that there's no replication of DNA, so interkinesis is pretty short. The cell is not trying to carry out its normal functions. We're just taking a brief break between meiosis 1 and meiosis 2 to grow the cell. We need to, because that cell went from a single cell to 2 cells and essentially halved in size. We're going to need to grow it again before we undergo another round of division. So interphase, we're going to grow the cell, and then interkinesis, we're going to grow the cell, and then we're going to move right into meiosis 2. In meiosis 2, we're going to see in prophase 2, chromosomes are going to condense. Those chromosomes are then going to move and line up along the midline of the cell. And then we're going to undergo anaphase 2. The centromere holding those 2 sister chromatids is going to separate. Chromatids of the new pair are now considered chromosomes, and those chromosomes are going to move to opposite poles of the cell. Once they move to opposite sides of the cell, telophase is going to occur. In telophase, nuclear membrane is going to form around those chromosomes at each end of the cell. Cytokinesis is going to occur in both daughter cells, forming 4 haploid daughter cells. And there we go. We have gone from a single diploid cell to 4 genetically unique daughter cells. The primary goal of meiosis 2 is going to be to separate those sister chromatids. Once we have done that, we have completed our meiosis. All right, so we're actually going to make another video where we're going to talk about when meiosis occurs, and specifically, we're going to break down the processes of spermatogenesis and oogenesis.